This method of separation that we are going to look at is called chromatography. Chromatography is the method of separating two or more components that dissolve in the same solvent. Some examples are pigments in plants, dyes in paints, as well as colorings in food products. Let us look at the procedure and the setup of the apparatus for chromatography. First, we need to draw a pencil line on a piece of chromatography paper, as shown in the diagram. Next, we place a drop of black ink on the pencil line, also known as the starting line. The black ink is suspected to be a mixture of red, blue, green, and orange dyes. So place drops of these dyes on the pencil line too. After we are done with placing the dye, we roll the paper into a cylinder and secure it, and place it into a beaker containing a suitable solvent for the ink and the dyes. The solvent will then travel up the paper and the dyes become separated. The chromatography paper can also be suspended from a glass rod such that the end of the filter paper is dipped into the solvent. The picture shows a chromatogram of dyes in different colored ink. When the solvent reaches the top of the paper, take it out of the beaker and leave it to dry. The chromatography paper with the separated component is called a chromatogram. This animation shows us the movement of the solvent up the paper and the separation of the dyes on the chromatography paper. Some points to take note. In paper chromatography, a suitable solvent for the mixture to be separated has to be selected. This is the most important point in order for chromatography to be successful. As the solvent travels up the water, the dye on the pencil line dissolves in the solvent. The more soluble the dyes are in the solvent, the faster they will move up the paper. Identical dyes will travel up the same distance in the paper. This is because they will have the same solubility in the given solvent used. Unknown dyes in the black ink can be identified by comparing the chromatogram obtained for the black ink with chromatograms of known dyes. Let us look at an example. From this chromatogram, the results of the experiment shows that the black ink is made up of 4 different dyes. 3 of the dyes are known. They are the red, the blue, and the green dye. There is no orange dye in the black ink, as it does not correspond to any of the colored spots that are found in the chromatogram of the black ink. There is one dye that is different from the 4 known dyes. Something to think about. Sketch on the chromatography paper how the chromatogram of a pure substance will look like. What will we obtain? If we have a pure substance, the chromatogram should only have one spot. This will mean that the substance is pure. So what are some of the applications of the chromatography? Government laboratories use chromatography to check that only approved dyes are used in food products. Chromatography is also used in the analyzing of the urine of athletes to check if they are using any illegal drugs.